And he is with us today, and this is quite an honor. Many of you have seen him on TV, as I have, and I'm going to share with you some information about him. But what I always think is important to know is I worked with Baylor for many years when I was an executive at the St. Luke's system, but I also worked at Baylor more recently as a consultant. And he has a reputation not only for being a great doctor, but a great guy as well. And so I always think that that's something that's really very important. So Dr. James McDeavitt serves in a dual leadership role. He is the Dean of Clinical Affairs at the Baylor College of Medicine and also serves as the Chief Clinical Integration Officer for the CHI St. Luke's Health System. In other words, he never sleeps because either one of those jobs would be enormously challenging and take up a great deal of time. His responsibility is to create linkages between physicians at CHI St. Luke's hospitals, between Baylor educational programs and communities in need of healthcare providers, between cutting-edge research programs and the bedside. All very challenging things, which he does very well. These linkages help the Baylor College of Medicine and CHI St. Luke's Health System deliver patient-focused, high-quality care to the people of Houston, of Eastern Texas, of the United States of America, and to the world. Dr. McDeavitt came to Baylor from the Carolinas healthcare system, which is now known as Atrium Health, one of the largest and most comprehensive systems in the country, where he served as Chief Academic Officer and Senior Vice President for Education and Research. During his 9 years in that position, he led a substantial expansion of the academic mission of that organization. His responsibilities included all aspects of the academic enterprise: undergraduate and graduate medical education, continuing medical education, research from bench to clinical implementation, and allied health education. Dr. McDeavitt also served as the founding associate dean for the Charlotte campus of the University of North Carolina Chapel Hill School of Medicine. And prior to his roles in the Carolinas, Dr. McDeavitt was the founding chair of the Department of Physical Medicine and Rehabilitation at the Carolinas Medical Center. As chair, he worked with a team that built nationally recognized education and research programs, while simultaneously expanding inpatient and outpatient programs regionally. So before I turn it over to Dr. McDeavitt, I hope everybody has a pencil and a paper, because he is ready, willing, and able to answer questions from us when he's through with his presentation. So this will be great because we'll really have an opportunity to ask those questions that we really want to know to someone we can trust who's a real expert in the field. So Dr. McDeavitt, thank you for being with us. The podium is yours.
Irene, thank you very much. That was a very generous introduction, and I'm sorry we're out of time. I'll try and keep my remarks to about 20 minutes because frankly, I mean, you all read the newspaper every day. You all see the reports in the media. You've lived and breathed COVID-19, so you know a lot already. So it's probably more useful for me to field questions from you than for me to tell you what I think. But I do want to spend a little bit of time just reviewing some of the more recent data and some of the science and some of the vaccine development and drug trials and that sort of thing. So I'm going to try and share my screen here. Are you seeing a full screen of my slide now? And, again, so trends, and I'll highlight, I put my Twitter address up there. I don't know if anybody tweets on this list, but I put it up there mainly because once a week, I have been, over the past 9 weeks, I've gotten into the habit of posting sort of a summary of what's going on with COVID-19 in the community, looking at the data, trying to think about the larger trends and patterns, and philosophizing just a little bit. But I invite you to follow me if you're interested in keeping up with that sort of thing. Y'all have probably seen this website at some point or another. This is out of Johns Hopkins, and I put this up just to remind me where we started. We are at now approaching 19 million global cases of this disease, over 700,000 deaths worldwide. And when I first got involved with this, it was last February, when we started a incident command center at Baylor to think about the dealing with what could become and ultimately did become a major public health crisis. But when I looked at this map way back when, there was a dot in Seattle, there was a dot in LA, there was a dot in Phoenix, and there was a dot, I think, in somewhere in Illinois or somewhere in the Midwest. That was it. So at that point, we looked at the map and said, gosh, I hope we can contain it and it doesn't come to Texas. Well, then you all recall we went through a period of time where we had the disease in Houston, but thank goodness it's only the people that are on the Egyptian cruise ship and don't have any spread outside of that. Well, that lasted for about a week. And then it became, oh, well, we've got community spread and we got to shut down the rodeo. And then our world changed. Then the NBA followed and you know the rest of the story. Well, now we are the US, ground zero of a global pandemic. A week ago, I would have said Houston, Texas was the epicenter of the global pandemic, with the possible exception of Miami, Florida, that was a little bit ahead of us. But just kind of stunning what we've all lived through over the past 6 months. And I'd like to go back and look at this every now and then just to keep that perspective. In terms of big picture, this is data that comes out of Harris County and the 8 surrounding counties around Harris in terms of the total patients that have come through hospitals. So this is an all-inclusive number. We're approaching now 19,000 people that have been hospitalized. That's a lot of people. And of those, 1,608 have died. This is as of yesterday. So again, a significant number of deaths. Part of the good news story is our mortality rate generally has been better than it has been in other parts of the country. New York City, in the height of their crisis, they had a mortality rate about 3 times what it's been in Houston, driven by a lot of factors that we can discuss. I want to sensitize you to the so-called R value. Has anybody heard of the R value? Talked about it. I'm sure it's been in the press, and some of you have probably heard something about it. The R value is mathematically calculated, and it's a measure of viral infectivity. And what that means is if I have the virus, statistically, how many people am I going to infect with my virus? And an R of 1 means that statistically I will infect 1 other person. An R of measles is highly contagious, has an R about 18. So 1 person with measles will infect 18 other people. So what drives the R? Well, part of it is just the nature of the virus. This virus is about 10 times as contagious as typical influenza, but not as contagious as measles. So when it started here in Houston, we had an R value back here in April of 2 to 3 or even more. So highly contagious. Well, then what happened? Then we started to become aware, we started to panic a little bit that our health system was going to get overrun. We had what we thought was a little surge back in April. It looks kind of quaint in retrospect, given what we then saw later on in the summer. But as we started paying attention to distancing, and as we went into work safe, stay home, work safe, and started shutting down the Houston economy, you can see the R value drop. And that's the other thing that affects the R value is sort of that social distancing. If I am infected with a highly contagious virus and walk into a room of 100 people, I can infect a lot of other people. If I am isolated in a room by myself, I can infect 0 people. My R would be 0. So clearly, masking, distancing has an impact on R. And that's what you've seen over the past several months. And this is a really important point. We got the R down to about 1. And when the R is less than 1, we actually see the viral load in the community start to shrink. That's where we want to be. If the R is greater than 1, we're going to see the viral load in the community climb. So I look at this number every day, And I'm really happy when it's less than 1, and I'm really depressed when it's greater than 1. And you can see it's oscillated. We did well in April and May, then it went up, then we got more serious about it. Then we had the 4th of July holiday, and following the 4th of July holiday, we saw it go back up again. It's come down and went up again. So it's as we pay better attention to distancing and closed bars and that sort of thing, that affects this R value. Really, really important number. So what does that drive? This is the summary of, again, Houston or Harris and the surrounding 8 counties of everybody who has tested positive for the virus on a day-to-day basis. So you can look back in April, this peak of 811 people, that's when we thought we had a real crisis on our hands, which again seems quaint. But we panicked back in. We looked at this and said, we could be the next Lombardy, Italy, or New York City. And we in the medical center did a ton of work to prepare to not be in that situation and not to spend a lot of time on that. But in New York, they did things like repurpose physicians to care for patients that the physicians really weren't qualified to take care of. There was an article in the New York Times. My daughter's a psychiatry resident. This resonated with me. There was a psychiatry resident that wrote that she was being repurposed to take care of patients in the ICU and manage ventilators, for which she was not qualified. And the article was about how guilty she felt about the care she was delivering to a patient. So way back when, we decided we did not want that to happen in Houston, and it did. I think we did everybody pulled together and did a good job. So then April and May were the good times. We sort of reached an equilibrium of 200 to 300 new cases a day. That's sort of a level that you could actually do effective contact tracing and maybe even control the virus. But then May 1st, we opened the economy a little bit. May 15th, we opened it a little bit more. We had a Memorial Day holiday. We had the George Floyd protests. So a lot of things happened. I don't think there was any one thing that caused this. We had a lot of young people in the community that thought this was over and kind of went back to their whatever young people do. But we got spread. So we started to see the case rate go up, and the case rate is still going up. Now, this is a little bit difficult to interpret because we're also testing a lot more too. So some of this is people that aren't terribly sick or maybe be asymptomatic, but we're clearly seeing the numbers go up and up and up. This is way in excess of what we can do with contact tracing. It's probably 10 times the volume we could do with contact tracing. And this probably still represents probably a fifth at best of the total disease burden in the community. So we're probably looking at something like 10,000 new cases a day in the metropolitan area, which is a lot of people. The good news in all this is, again, we panicked in June because look, we're having a terrible surge. Here we go. Right on clockwork, the hospital census started to go up. Well, now, even though the case rate's going up, for the past 3 weeks, the pressure in the hospital has been going down. We've seen less admissions in the hospital. That's a really good thing. So I think we're sort of past the most recent wave. We need to maintain our vigilance, and I'll come back to that in just a second. Other things to touch on, I'll cover this very, very quickly because I think it's sort of old news at this point. All of you are involved in some sort of business. All of you are probably junior epidemiologists and experts on public safety at this point because most of you have figured out some way to safely open businesses and think about how you're going to do that and protect your employees and protect the public. I would say fundamentally the most important parts of that are monitoring symptoms, making sure that whoever is coming into your place of business, do they have symptoms, do they have a temperature, keeping sick people from coming into your environment. I think that's probably the single most important thing you can do at this point. And obviously cleanliness and appropriate screening criteria. We have this guidance posted on the Baylor website. If you're interested in more detail, I'm not going to cover this in a great deal of detail today. We can take questions after. I'm going to focus for just a second on Baylor as a business because we take care of patients. We generate our revenue from patient care. And the beginning of this graph is our daily patient volume. Which you can see when the epidemic hit with the vengeance, we lost virtually overnight about 75% of our volume, which is tens of millions of dollars, and still have doctors that need and want to get paid through all that. We did a couple of things really well. One is we were able to create a telemedicine platform to create video visits and do that very quickly. Now, full disclosure, we'd been working on that already, coincidentally. So we were sort of queued up from a regulatory and billing perspective and done a lot of the advanced work. But all this orange color here is volume we could replace through a telehealth strategy. And then you can see over time, we've started to recoup both our physical volume and our virtual volume. So it's been a nice success story. I would emphasize that if you need medical care, one of the negatives out of this whole pandemic has been people not seeking care that they need. So we've seen bad outcomes from heart disease. We've seen bad outcomes from stroke. If you have a serious medical condition, you should still get your care. I would say if you have conditions and just getting your regular screening tests done, you should still be following up and getting your screening tests done. And I can tell you that walking into a clinic at the Baylor College of Medicine today is probably the safest place in the city you could be right now. So that's I would not hesitate to get appropriate medical care. Testing, I get a lot of questions about testing. There are lots of options now. It's been in the press a lot because some of the results take some time to get back. Many of you have testing set up through your businesses or your occupational health programs. Most of you can probably get testing through your primary care doctor. They can order it. They can do it through commercial labs. I would say if you don't have access to testing, one thing you can do is access Baylor. Again, I'll give you these slides and you'll have all the information. But we can schedule a virtual visit with a physician. We have a parking lot operation, scheduled an appointment. You come in, you get swabbed, never get out of your car. I was down there last Wednesday swabbing people's noses for half a day. And we got our results back usually the same day and almost always within 24 hours. That's we've now tested over 10,000 people. A little bit about the science, because the big story now is the vaccine. When is the vaccine coming? When is that going to be our salvation? And to think about what a vaccine does. What a vaccine does is prime your immune system to fight off a foreign invader. And when you sensitize your immune system with a virus or with a piece of a virus, initially you get this uptick in the antigen loads. Your body sees something that it sees as foreign. It responds to that initially about 7 days later by creating something called IgM, which is an immunoglobulin. It's specific to that particular antigen. There's about a 2-week period where you see the IgM peak and then it goes back down again. And then your body starts to produce the red line, which is IgG, which also fights off the virus, but has a longer term effect. So when you hear about vaccine studies, one of the things that you hear people talk about, well, how durable is that effect? Part of what they're talking about is measuring this IgG days to weeks out from the vaccination. How long do you keep those antibodies in your blood system, which gives you some idea of how long the effect of the vaccine is going to last. Not entirely, because you can actually have really, really low levels of antibody and still have a degree of immunity. So this is what the vaccine is going to do. The other thing you hear a lot about is the so-called phase 1, phase 2, phase 3 trials and Operation Warp Speed and trying to speed the vaccine to market. I would point out in the past, the fastest we've ever, as a country, been able to produce a new vaccine was for the Ebola virus. Now, to remind you that that crisis, which was, when was the Ebola crisis? The early 2000s, or if anybody knows or remembers off the top of their head how many people were affected by Ebola nationwide, I think the answer is 9. 9 total people got our attention, build a virus. It took 5 years to get that virus into production, long time. Now, what Operation Warp Speed is really doing is using a lot of federal money to fund a bunch of different virus trials simultaneously, and then in a stupid way economically, but from a smart way from a public health perspective, building infrastructure to mass produce all these viruses in advance of actually knowing if they're going to work. So we're producing a lot of vaccines and already manufacturing the vaccines and spending tens of billions of dollars on that process, knowing that some of these bets aren't going to pay off and we're going to be just wasting money. But that's an effort to get the speed it to market. The science of this is that when you test a vaccine, you start testing in animals to make sure it doesn't do something terrible to an animal, to make sure it's safe. Then you go into phase 2 trials, which are very small groups of people, literally dozens of people, that get small doses of the vaccine, again, to look at safety, and to start to look to see if you have a mounted immune response. Phase 2 are larger groups of people. And in phase 2 trials, you start to do things like give different doses of the vaccine and measure the antibodies produced to see what the ideal dosage is, and you have to do split dosing. So all that occurs in phase 2 trials. And then finally in phase 3 trials, which is where now the Moderna trial you've heard about, we're doing it at Baylor. In this green dot down here, where are we in the phase 3? These phase 3 trials, the 3 over here, that Moderna is one of those trials. That is a trial where you test it in thousands or sometimes tens of thousands of people in real life situations to see if it actually confers protection from the disease. So you have to get through all those steps before you say I have a safe and effective vaccine. And I will say the Moderna trial, that trial will go on for 2 years. And it goes on for 2 years to make sure that the vaccine doesn't cause some safety problem, like swine flu. Some of you probably remember swine flu, which causes a neurologic problem. Well, sometimes it takes weeks to months before you know if there's going to be a bad outcome from a vaccine. At some point, we're going to have a vaccine that's been through phase 3 trials, and the FDA is going to make a decision that we think this is safe enough to fast track to market. That's sort of the story of vaccines. We've got 140 molecules and proteins that are out as possibilities. And you can see the numbers in phase 1, phase 2, phase 3 in terms of getting those approved. Now, the vaccine is kind of our great hope for the future. Realistically, I think it's likely we'll have a vaccine by December. When we have that vaccine, it's going to have to be produced, distributed, we're going to have to get it to people. It's going to take, in the best of possible circumstances, 3 to 4 months for that process to occur. So if all the dominoes fall exactly as we want them to fall, honestly, it's going to be spring at the very earliest before we get relief from the vaccine. So part of my message to people has been masking, distancing, doing all these things to control the virus, to keep that R value down is critical if we're going to go back to school and back to work and open the economy and resume some semblance of normal life. And the vaccine is not coming to save us in the short term. It's a longer term play. I think I just got a couple more slides. I'd highlight one of the fortunate things about having the Texas Medical Center here and Baylor College of Medicine and others is that we are involved in all the major research projects. We can talk about, highlight a couple. One, the hydroxychloroquine, which I don't know if anybody watches the news on the call, but you know, hydroxychloroquine has been in the news. It's controversial. I will tell you, as of today, there is no role for hydroxychloroquine in the management of COVID-19. In spite of the hype and what you hear, there is excellent randomized controlled clinical data that shows that for sick people, it doesn't help, and in fact, actually causes some cardiac problems. Small, but real. So first, do no harm. Doesn't work, hurts some people, don't use it.
It's one of the things I signed up for to watch.
The second issue, can I rule out the possibility, somebody's not on mute, so we're hearing your conversation.
Called hydroxychloroquine or whatever.
Please
he
mute.
Said, I'm here to tell you it plays no role.
Which is probably more interesting than my presentation.
We can hear your conversation, so please mute. Thank you.
Now, in the interest of full disclosure, can I say definitively if somebody took hydroxychloroquine prophylactically, they don't have the disease, they're going to take it once a week to try to prevent the disease, could that work? The answer is possibly. The trial has not been done to prove that or disprove it. But as of now, the science would say there is not a role for hydroxychloroquine in the management of the disease. There's a second drug called remdesivir, which is an antiviral agent that has been subjected to randomized controlled clinical trials, some of which are being done at the Baylor College of Medicine, that shows that it does shorten the hospitalization by about 4 days for really sick people. It does work. It has some efficacy. It's not the penicillin of COVID-19, but it has some positive impact. You've read about steroids now. Steroids have been proven to have some impact, and they're dirt cheap and relatively safe. And then there are a number of other drug agents we could talk about that are in trials. So we're getting a handle on things that we can use to effectively treat the virus. The last thing I'd highlight, also going on at Baylor and in St. Luke's and Methodist, is a lot of work on the convalescent sera trial. Convalescent sera is a more than a century-old technology. It was used in the Spanish flu, where you infect people, or in the case of the Spanish flu, horses, with the disease. You take their sera, you purify the antibodies from the sera, and then you give those antibodies back to sick people. So we do that with COVID-19 patients. If you've had COVID-19, you can go to the blood bank, donate plasma, and then give the plasma back to other people. That has been proven to have some efficacy in sick people, and increasingly there's some trials underway on trying that for less sick people. So I'll finish with, and I've already sort of touched on this, well, what to expect in the future. Let's reflect on what we've learned so far here in our Houston experience. We had a surge in April. We got on the back end of that surge into May. And then we sort of patted ourselves on our back and breathed a collective sigh of relief and said, boy, that was tough. I'm sure glad we beat COVID-19. Well, we didn't. All we did was distant and get people separated from each other. It's still out there in the community. And I think about it like a pile of dried kindling. If you've got a pile of kindling that's all bunched together in a pile and a spark hits it, the whole thing goes up very, very quickly. What we do with social distancing is we take that pile and we spread it out so the sticks won't catch each other on fire. But as soon as you push them back together again, they reignite. So as long as we have a population that is not immune, as soon as we start pushing that population back together again, we will get another surge. So we've been through a surge in April, lessons learned. We then had a surge that was literally 8 times as bad as the first surge, and we were at serious risk of overwhelming our healthcare system. It looks like now we're past that second surge. I guess my plea, and I don't know how to do this, is we have to take this seriously until we actually have this vaccine, which could be 6 months, could be a year from now. Masking, distancing, avoiding aggregate situations, not having, the bars are closed. Now kids are going out and having parties in their house and having 50 people over to their house, and we're going to specs. I mean, that's just as bad and maybe worse. We really have to figure out a way to take this seriously. And then second point is, I think what to expect is when you look at those sort of curves that go up and down over time, and we'll continue to see that. We will eventually open schools. We'll eventually start to let bars open up again, I imagine. We're going to have a Labor Day holiday. We're going to have a flu season in the fall. There will be lots of things that happen that are going to stress our community. And we'll get more surges. So I think we're going to see this tension between shutting things down, distancing, and opening up. And we're going to see the virus sort of bounce around as we find the sweet spot going forward. And we have to find that sweet spot, because the extremes are shut down the economy, put everybody out of work, really hard cost, personal cost of doing that. But great from a public health perspective. To the other extreme, let's ignore the virus and we'll go back to business as usual and we'll have a bustling economy. Well, the reality is neither of those things are appropriate. And good people can disagree about where the sweet spot is, but I think over time we're going to find what the right sweet spot is between those two extremes. I think that concluded my prepared remarks, and I'm happy to take any questions.
Hi, I have a question. Can you hear me, or can you see me?
Yes, I can hear you.
Okay, cool. Hi, it's Anna. So thank you so much. I really enjoyed everything that you presented. So I'm gonna ask about, a little bit more about the vaccine. So I understand the phases. I'm very familiar with the clinical trials, and I think a lot of people are too optimistic, to be honest. But anyways, one of the ones that I have a question about is the famous mRNA one. So to my understanding, mRNA vaccines have only been seen in animals as of now. Is that still true? That's still true, correct?
Yeah, so it's a good question. So historically, when you look at making a vaccine, polio, for example, you're either taking the virus and killing it, and then injecting it into people, and you react to the killed virus,
Mhm.
or you're taking a weakened and attenuated version of a virus and injecting it. And what is being done in this case is the RNA vaccine. There have never been RNA virus vaccines before. It's taking the genetic material of the virus,
Right.
and creating antibodies to that genetic material. So that there are two problems with that. One is it's sort of an unproven concept. You're absolutely
Mhm.
right.
Mhm.
The second is RNA as a molecule is incredibly unstable.
Yeah.
So this could be a solution for Houston, Texas.
Mhm.
It is not a solution for rural areas, for impoverished areas, for third world countries, because it has to be handled carefully, refrigerated. It's not very durable. It's a very unstable vaccine.
Yeah, and that's what I was wondering, because I mean, also one of the things that came up to my mind is like RNA is quite unstable. And so what you're saying is that to mass produce where we can get it to everyone, to a whole global population, let's say, would be almost
I think it's impossible.
right. Okay.
An RNA vaccine is not going to be the salvation of the planet. There's going to be another solution to deal with sub-Saharan Africa.
Right. And then one last question, I guess. Does the RNA code the antigen, or so our body can detect it and then produce the antibody? Is that what it is, if I understand it correctly?
You know, viruses are really evil creatures. I mean, typically a bacteria. When you get sick with pneumococcus, when you get pneumonia from a bacterial pneumonia,
Mm-hmm.
Bacteria has a cell wall. It's got a nucleus. It carries its own genetic material. It's got mitochondria. It has all these cellular functions. It is a living organism that reproduces. Virus is like a stupid bag of genetic material. It is a cell wall, and contained in that cell wall is genetic material. It has a protein on it that allows it to attach to a human cell. It injects the viral material into the cell, and it hijacks the workings of the cell to replicate the proteins necessary, including RNA, to replicate the virus. And then the cell explodes, all the components extrude, and the viruses self-assemble and create millions of more viruses. So it purely exists to replicate. That's all it does. I got kind of distracted. I forget your question now, but that's the.
No, I guess I was asking, and I know I'm talking about vaccines, but I'll be presenting on this in a little bit later in the month. So I guess my question was, the mRNA that's injected, is it the genetic material for the antigen or the protein coding of the virus, and therefore our immune cells are able to detect, oh, this is a foreign invader, as you put it, or right?
Yes, it is the RNA that comes from the virus.
Gotcha. Thank you.
Lisa, you have a question?
I do have a question. Thank you, Dr. McDeavitt. There are two, actually, it's kind of almost two questions. On your chart, there was one green dot that said something was ready. One of the vaccines, there was one that sort of was in the ready to go, which wasn't mentioned. And I don't know if that meant anything or not.
Yeah, I see what you're looking at. That to be completely honest with you, I don't know what that is.
Okay.
I just made this slide yesterday, and I don't recall. There is nothing in the United States that is currently approved. That could be that there's been an approval worldwide, but there's nothing approved in the U.S. right now.
So my second backup question is, when will you take this vaccine?
That's a really good question. I will probably take it when it's available.
Okay.
And I have the bad things that happen from vaccines are rare and unusual enough that I think the likelihood of having a swine flu, which was in the 1970s, that having that recur is very, very small. And I think the benefit to getting this pandemic under control, which is affecting millions of people and devastating our economy and threatening our way of life, I think in this case is worth the risk of taking a vaccine that perhaps has not been through each and every safety check that we would like.
Thank you.
That's going to be a little bit of a personal choice. So build on to that. The problem is the anti-vaccine movement out there. There are some studies that show that fully 20% of people do not intend to take the vaccine when it's available. And there are people that have, I think, misguided but philosophical issues with the vaccine. And then there are going to be others that are going to say, this hasn't been fully tested, I'm not going to take it. So we're going to have to get to a point that 60 to 70% of people take the vaccine before it's going to have an impact.
Thank you.
And Beth, I think you have a question. I think he's answered some of it, but do you have another part, Beth Young? In the chat, she was talking about the vaccine and of how long you think it would take to inject a large portion of the population. I think that you had answered that, but how much of the population needs to receive it?
Yeah, nobody knows, but the estimate is at least 60%, and it could be more than that.
Wow.
Well, as a follow-up to that vaccine question, I've heard that a vaccine will be approved if it is at least 50% effective.
That's correct.
So if a vaccine is 50% effective, then how safe can we be resuming, going to restaurants, going to movies, having large conferences, and so forth?
Yeah, this is, it's a difficult question. Let's look at the Spanish flu experience. And if anybody's interested, I don't know if anybody read the book by, who wrote it? Eric Lawson, I think. The Great Influenza, about the 1918 Spanish flu epidemic. Really, really interesting, and a lot of interesting parallels to today. Well, way back in 1918, the concept of a virus had not been developed yet. They desperately looked for the pathogen and couldn't find it. They didn't find the pathogen for the Spanish flu until 15 years after the Spanish flu was gone. It took 2, or some people would even say 3 years for that to sort of wash itself out of the population. So eventually we'll survive this. I mean, it will pass. It might take years for it to pass. We'd like to accelerate it. So in terms of the vaccine, I think the vaccine helps to accelerate that process, and it could do that in a couple of ways. We talked about the need to vaccinate 60 to 70% of people. Well, another strategy would be, let's go into nursing homes and congregate living facilities and vaccinate those people we know are going to be at very high risk. That would be a way to help to slow the spread. Getting back to my stick analogy, when you have that pile of dry twigs, if you ever try to start a fire, all the dry twigs go up like dry sticks. But if you start to mix in some soggy sticks and some green sticks and the ones that don't catch quite so, then the spread is slower. And we're still going to have a lot of people get sick. We're going to have a lot of people get the virus, but hopefully we don't all get it at the same time and overwhelm our healthcare systems and fill up our ICUs. So I think that's part of the value of the vaccine, is not necessarily eradicating the disease, but sort of slowing the spread so it spreads more gently across the population over a longer period of time. There's also some possibility the vaccine should help to decrease the severity of the disease. So like flu, sometimes your flu shot doesn't keep you from getting the flu, but it keeps your symptoms from being as severe as they would have if you hadn't taken the vaccine.
Right. Well, I think, Teresa, you have a question? I think you're on mute, Teresa.
Thank you for letting me know that. Sorry. Hi, Dr. McDeavitt. Thank you very much for your presentation. And by the way, my son also last year finished his residency in psychiatry at Baylor College of Medicine.
Oh, excellent. What's his name?
His name is Mark Urowiz. He just joined. He's now working for you at Menninger. So.
Outstanding.
Thank you for your presentation today. My question is, what does 50% effective mean? Does it mean that 50% of us will be protected, or that it reduces the chance that we'll get the the virus by 50%.
What 50% means that in phase 3 clinical trials, when you're administering the vaccine to thousands of people, and you know how many people should get the disease in that cohort of people, that the new incidence of disease would be reduced by 50%.
Thank you.
And Ashley?
Yes. Hi, Dr. McDeavitt. It's been a long
Hey, Ashley, how are you?
I'm good. Nice to see you again. You talked about the inpatient clinical treatment trials that y'all are doing. Could you expand on any of the outpatient treatment trials that y'all are doing and what you're seeing there? I mean, as we talk about, and I agree with you, I think vaccines are going to be delayed. What do you think from an outpatient treatment perspective?
To be perfectly honest, I don't know that we're engaged in a lot of outpatient therapeutic trials. We're engaged in a number of trials looking at disease prevalence and disease spread and sort of long-term sequelae of the virus, but not so much in administering medications to people to try to prevent.
Linda Avedon, do you have a question? You're on mute, I think. Linda, you're on mute.
Now.
We can hear you now. But the connection is really bad. So I'm then it's of the number
is how many?
Linda, your connection is really bad.
Yeah.
I'm going to read it for you.
Can you just
I think we can't hear you, but I see
you see it in the chat.
The question is, if I read it correctly, of the number of deaths, how many have died of other conditions?
Thank you.
Such as cancer, heart disease, et cetera, versus COVID directly. Don't know that number off the top of my head. Not sure we fully understand what that number is, but we do know that we have seen a significant decline in people seeking care. I have not seen published data on the increase in cardiovascular mortality yet. I think it's a little bit early to know that definitively, but I think it's going to be consequential. What'll it be? So it's easy to sort of get at. I have stroke symptoms. I choose not to go to the emergency department. Therefore, I don't get my clot busting drugs quickly enough, and I end up having a worse stroke. I think we'll be able to answer that question fairly soon. A little harder to think about. I didn't get my mammogram. I didn't get my screening colonoscopy. I developed a cancer that ended up being more advanced before I sought treatment. I think it's going to take several years to figure out what that impact has been.
Wow. So we're talking about someone that has had COVID and perhaps did not even know it, didn't have symptoms or whatever. Are the antibodies that you develop, is it important for us to go get tested to see if we have antibodies? Or and also, are you immune? I know we have a good friend on the call today that's had the disease and wondered, is she immune now, like having a vaccine?
Yes, so if you have had the disease and you're interested in donating your plasma, we actively accept plasma donations. When you go to donate plasma, they check you for antibodies. And in some cases, many people do not have antibodies, and they're not, their plasma can't be used for that purpose.
So you're pretty safe of not catching it again then if you...
Well, the honest answer to that question is nobody knows. The experience has been with other viruses that typically once you are exposed, you are immune for a year to two, and in some cases much longer. But there was some suggestion early on that people were getting COVID and then getting it a second time. I think most people think that's probably not the case at this point, and we also found that people were shedding this viral RNA, which gives you a positive diagnostic test for a long time, even though they were having no symptoms, not really infected. So it's I don't know the answer to the question. Nobody does, but I suspect you have immunity for a year or more if you've been infected.
And one more question about the mask. You hear about, certain masks are not as strong as others. Does it really matter? Your mask, does it need to be a 95 or whatever the.
Yeah, so in terms of masks, there are really two different classes of masks. They're the so-called N95s, which are famous. They're PPE, and those are the ones we're in terrible short supply. The openings in N95 are small enough that they can actually filter out viral particles, so that protects you from infection from the virus. That's what healthcare providers wear when they're up in people's faces or doing intubations or for maximum PPI, PPE. The virus can also enter through your eyes. So what we're doing now is N95s plus a face shield to protect your eyes. That's if you are dealing with somebody, dealing with body fluids, dealing with coughing and fluids, etc. That is different from a face mask. Face mask is usually a multiply piece of cloth that doesn't filter virus, because the virus is small enough it gets through the holes in the cloth. But it does filter droplets. When you sneeze or cough or breathe heavily or exercise, you expel droplets containing virus, and the mask helps to slow and decrease the number of droplet particles and slow the spread. Though I said that the mask protects you, not me, there's some evidence here recently that the mask actually confers a little bit of protection on the wearer as well. But it's not really PPE. But it does help to slow the spread.
Spread down. Wow. And Anna, did you have another question?
I did. So I'm going to go back to, you compared very briefly New York and Houston. And so I kind of want to talk a little bit about that or ask some questions. And I usually word vomit, so sorry if I'm vomiting words. So one of the things that I find interesting, right, is that Houston kind of experienced an increase in cases later, so we were kind of more ready to fight, if that makes any sense. And so New York got it very early on, and so they were just rolling with the punches. And so what kind of things is TMC, the Texas Medical Center, doing in preparation for those waves that we are going to have, that we for sure will have, as we find the sweet spot?
Yes, so that's another really good question. Actually, I referenced my little Twitter feed. I wrote about this probably 6 weeks ago
I'm following you.
So. Is that when we started to see the second surge, we had a lot of people in the hospital. I had doctors coming to me and saying, we're seeing a lot of patients, but they just don't seem like they're as sick as they were. So we started asking ourselves a little bit, why are outcomes better? And things change fundamentally too. So 2 periods of time, length of stay in the ICU when we first started this in April was about 18 days for a COVID-19 patient. Length of stay in the ICU now is like 5 to 6 days. So a dramatic improvement in length of stay. Your chance of winding up on a ventilator, admitted to a hospital, your chance of winding up on a ventilator back in April was about 43%. Your chance of winding up in a ventilator today, if you're going to a hospital, I think is about 20%, or it's about half what it was back then. So dramatic changes in the way patients are managed. The question is why? I think part of the why is we have some drug treatments that we didn't have then. Back in April, we had no clue. We had absolutely nothing to do that worked. We actually used hydroxychloroquine back then because it was the only thing out there that had been suggested to have any utility. Second, this is a strange virus. The patients react in different ways than typical patients with pulmonary issues. So they go on ventilators. People did very poorly, and we discovered some things like, well, we didn't put patients on ventilators as early. We left them off the ventilators. We started using high flow oxygen, which people seem to tolerate pretty well. So we changed treatment paradigms. We discovered in ARDS, another condition, that when you ventilate people face down, which is a really unnatural way. Think about how complicated an ICU patient is and all the wires and tubes and everything hanging off of them. Imagine then taking that patient and putting them so they're facing the floor instead of facing face up. Not easy to do, but it's easier to ventilate these patients when they're face down. So we implemented a lot of prone ventilating protocols. I think part of the difference is people got better at it. It became more routine. I think that was a piece of it. I think part of it was we were panicked. There was no PPE back in April. Now we got lots of PPE. So healthcare providers aren't concerned they're going to get sick and take the virus home to their families anymore. They were back then. I think we saw a change in the population to a younger population and an older population. I think that's made a difference. So I think it's been a lot of different things.
Yeah.
And I think the other thing is we had time to prepare. We looked at New York. I mean, New York was New York, kind of out of the blue. It sort of took them by surprise. We've been sitting here for a couple months looking at their experience and trying not to be New York. And I think that's helped.
Yeah, cool. Thank you for answering. I know that there's a lot of factors that go into it, but I think that a lot of people fail to realize how healthcare providers and researchers are learning as they go, and usually have like 10 years of data to deal with the disease. So thank you.
There was another question about antibodies and immunity, and it said that if they've heard that you can't detect antibodies after 90 days, then how can you be immune for 1 to 2 years? Not all that unusual not to detect antibodies until have immunity. You've got cells in your body that are primed to fight a particular antigen that go and live in your bone marrow. The antibodies can be there in undetectable levels. When you're exposed to the antigen again, your body is primed to very rapidly mount an immune response.
That's great. We have time for one more question. Or we'll let Irene, we've kept you now nearly the whole hour. You can see how intriguing your speech was and how much we're wanting to learn more, about the disease. So Irene, I'm going to hand it over to you.
I signed up for the full hour because I wanted to earn my pay.
Okay. Well, I think you did. Thank you.
Well, thank you so much, Dr. McDeavitt. I think this has been just a very interesting discussion. It's so nice to be able to talk to somebody that's not running for election or not have to be concerned about what they say and what their boss thinks. But it's really just great to have your straightforward, frank answers to all of the questions. And I think it will enable all of us to know what to do to remain safe, because I think we're all trying to remain safe, and it's very helpful to hear. And it's also very helpful to hear your positive viewpoint on this is going to end sometime, even if we're talking about next spring. We all plan to be around next spring to see it. So that would be very nice. I'm hoping to have some fun next summer and travel more and all that good stuff. So I hope all that happens. But thank you.
We will definitely get through it, and I appreciate your time. And I would just, ask this group, you are ambassadors, you are representatives, people pay attention to what you do, and you've got employees that you influence. So if we can just keep getting the message out there to maintain our vigilance, that we will get through this. Again, this too shall pass.
Thank you so much.
Thank you again. We'll bring you back, I'm sure, for an encore performance.
Thank you for having me. I'm going to sign off now. So take care.
Thank you again.
Yeah.
And I know Irene wanted to welcome all of you personally and tell you a little bit more about what's going on with the chapter and the chamber, and also hear from you. Anything you'd like?
I would, we like to do introductions, but I do want to pass along something. There is a program that has been started in Sugar Land, where if you buy a gift certificate from a Sugar Land business and apply through the Economic Development Council website, they will give you a gift certificate of 50% to use at another Sugar Land business. And I believe that they also then provide a gift certificate to our first responders. So it's really,
wow. We need to put it in our newsletter.
Yeah.
Can you send us a link for that? And
I'll send you the link, but you can always go to the Sugar Land or the Fort Bend Economic Development Council.